You drop a single synthetic tea bag into a mug of hot water, expecting a simple morning ritual. As the tea steeps, the heat breaks down the nylon, releasing microscopic fragments directly into your drink. This diagram illustrates the shedding rate in real time. That one tea bag releases 11 billion microplastic particles into the cup. If you prepare formula in a plastic baby bottle, hot water releases 55 million particles per liter. For decades, we pictured plastic waste as a distant, visible problem, bottles washing up on remote beaches or drifting in open ocean currents. The pollution has crossed from the environment into our homes. Through our daily meals and morning routines, this synthetic material breaches biological barriers and settles deep within our organs. In 2018, a team of researchers tested human stool samples gathered from volunteers across Europe, America, and Asia. These were people with vastly different diets and lifestyles. This map tracks their results. The data reveals that every single sample, without exception, contained microplastics. Finding this indestructible material inside everyone they tested forced scientists to ask how it gets inside us, how it breaks down, and what it does to our organs. Plastics are built from long, tightly bound chains of molecules called polymers. When exposed to sunlight, UV rays and oxygen attack these chains, snapping the bonds and causing the material to turn brittle and crumble. Rain and wind scatter billions of these microscopic splinters across agricultural soils and fresh waterways. They are absorbed by crops and ingested by livestock, guaranteeing that the food we eventually eat is preloaded with synthetic fragments. Once inside the stomach and intestines, the constant influx of particles disrupts the gut microbiome. The plastic alters microbial activity, creating an imbalanced state called dysbiosis. Lab tests show this disruption elevates chemical markers closely tied to digestive tract disorders. Patients suffering from chronic intestinal inflammation and Crohn's disease consistently show higher concentrations of microplastics in their colons. Our digestive tract acts as the first line of defense, but it is failing to block the continuous wave of plastic passing across our plates. We also absorb plastic through our lungs. Every time we wear or wash synthetic clothing, we shed countless microvibrators into the air. We take about 15 breaths a minute at rest, pulling these floating particles straight into our respiratory system. By flushing the lower airways of patients, pulmonologists have tracked these fibers down into the alveoli, the deepest, most delicate regions of the lung where gas exchange occurs. The physical size and chemical durability of these plastic fragments closely mirror asbestos fibers, a known hazard that can penetrate deeply and stay lodged in lung tissue indefinitely. Patients with the highest concentrations of lung microplastics experience noticeable airway constriction, worsening severe respiratory conditions like asthma and COPD. This diagram illustrates the scale of a newer, more insidious threat. By zooming in past standard microplastics, we find nanoplastics, particles so tiny they are up to 50,000 times smaller than a single human cell. If synthetic waste is light enough to drift on air currents and travel to remote beaches, it is small enough to circulate freely through human veins and arteries. Researchers examining maternal tissue have located nanoplastics in the amniotic fluid, as well as embedded within both the mother's and the fetus's sides of the placenta. Because they are infinitesimally small, nanoplastics slip right past our tightest biological barriers. We are carrying a synthetic burden before we take our first breath. When these particles invade our tissues, our immune sentinels, called macrophages, try to eliminate them. The cell detects the intruder, wraps its membrane around the plastic, and pulls it inside to digest it. But commercial plastics like polystyrene resist chemical breakdown. The macrophage fails to digest the particle, and in a desperate attempt to destroy it, continuously pumps out hydrogen peroxide. Hydrogen peroxide floods the tissue, causing oxidative stress that breaks human DNA strands. This recurring damage makes the plastic genotoxic, forcing genetic mutations. In tests, mutated lung cells didn't die. They penetrated tissue layers, behaviors identical to spreading tumor cells. This active chemical intruder drives mutations that make it highly carcinogenic. We are passing this cellular damage down. Medical researchers are now maintaining massive biobanks of umbilical cord blood and placental tissue to track how early exposure impacts a child's long-term cognitive and physical health. Meanwhile, global plastic production is aggressively expanding. Projections indicate the total volume of single-use waste could nearly triple by the year 2060. We must sharply reduce our daily reliance on these synthetics, or risk permanently altering the biology of our own species.